Good morning, good afternoon, good evening, good day. And so today we're going to pick up somewhat where we left off, and we're going to keep moving through the incredible diversity of arthropods in our overall voyage through the Dunn et al. Phylogeny snapshot of all of animal life. We're going to talk about the myriapods and the crustaceans today. And so we're going to be jumping between Brusca chapter 22 and chapter 23. When we were last together, we were talking about, we dipped our toe into the hexapods. We're going to come back. But we talked about kind of the synapomorphies, the unique things that make this group really cool. And then we talked about entognaths. The colembla was the poster child for that. And the ectognaths, the insects, that is a synonym for insects. So these groups where the placement of the jaws inside of the mouth or recessed inside of the mouth or outside essentially define what you're eating and who's related to whom. Then we talked about a lot of the different aspects involved in the diversity, engines behind the diversity of hexapods, of insects. Some of these engines involve their mouth parts and how all of these different parts moving together in synchrony actually allows a myriad of successful adaptations of who's eating whom, when, and where. From herbivory ancestrally to sucking in bloodsuckers and parasitoids and all sorts of just a vast diversity of feeding strategies that exploded onto the world with the hexapods. We talked about how their development and how particularly the holometabolist form of development where the juveniles and the adults occupy really separate niche space, accentuated and probably accelerated diversity because they weren't competing with themselves any longer. We talked about molting and how one of the consequences of molting, this thing which helps diversity, also involves some really stressful periods in life. And we zoomed in today to thinking about respiration and how having your tracheoles and your tracheal system kind of ripped out with each molt is likely very, very stressful. We talked about the close relationships in terms of insect diversification through geological time with fungi initially and plants secondarily, and then co-evolved relationships with plants, flight, eyes, and vision, just lions and tigers and bears. Oh my. Where are we going today? We're going to talk about the myriapods, which are, you know what these are. These are the centipedes and the millipedes. And the crustaceans, like food, for much of the surface of our planet, strangely called Earth, which should have been called ocean. So we'll talk about the synapomorphies and predatories, relationships with the chylopods, that they are predators. Those are the centipedes. With the diplopods, with the millipedes, we'll talk about their synapomorphies and how their role in a lot of ecosystems is really as prey. They are detritivores and herbivores. And so there's a whole bunch of incredible adaptations they have associated with defending themselves from predators. And then we'll jump into really just literally, I suppose, an amuse-bouche, just a little taster kind of menu of the crustaceans. Crustaceans is not a monophyletic group. The only way to make crustacean monophyletic is to say insects and crustaceans. And there's an incredible number of great stories and thrilling kind of adaptations spread across this world. So we're just going to dip into a few of them and talk about, in general, some of the adaptations that characterize this group. We'll talk about the Yeti crab. We'll talk about how everything, if left to itself, seems to become a crab. We'll talk about their reproduction and then some adaptations that we've seen pop up associated with, just as you focused on in lab, moving into the terrestrial environment. So the bigger picture learning outcomes that I have in mind here is to think about how they are feeding and defending themselves, and thinking about that kind of transition to terrestrial, and stresses associated with being terrestrial when you are restricted to very, very moist environments. These three themes kind of capture most of the examples that we'll talk about today. So here we have the myriapods in yellow. We've got chylopods and the diplopods. Those are the groups we're going to focus in on, but these are the centipedes and the millipedes here in yellow. And then we've got the crustaceans. And you can see if you go back to the root of this tree that there's a star here indicating that it is not a monophyletic group. And the only way that crustaceans become monophyletic is if we're going to include the insects. More on that later. Let's drill down to these groups, the centipedes and the millipedes. I have a particular bias here. I'm team millipede, and that'll become apparent. So what are some of the synapomorphies for the myriapods, the centipedes and the millipedes? That there's segmentation, but no tagmatization, not to the same degree as we see in the insects. So the trunk's not divided into a thorax and an abdomen. Only one pair of antennae. That the median ocelli, this kind of really simple light eye, or eye associated with light, has been lost. The compound eye is really simplified. It lacks crystalline cones. The ommatidia are far and few and loosely arranged. These are dark things that are adapted to not living in direct sunlight and not really depending on vision a lot. The spiracles cannot be closed. They have gas exchange with a tracheal system, but the spiracles can't be closed, and that leads to a really tight dependence on a moist environment. That if it becomes dry, many insects can close off the spiracles and stop arrest water loss, but these ones cannot. And then they have these kind of crazy, beautiful organs on their head called the tomishvari or the tomoshvari, and these are probably associated with that dependence on tight association with moist conditions because they might be chemo or hygro receptors, but in truth, their exact role is uncertain. Here's a restatement of that. So that's me kind of like distilling some of what is in box 23A, the subcharacteristics or the really cool things about the myriapods that Brusca et al. Have talked about. So I offer it up here as a reminder. This is a great way using these boxes inside of the chapters of the text, and the images figures that I pull when I use them here, use them as a way to jump around inside of what is admittedly a very long text. So here's the tomoshvari. So this is their paired sense organs. So we're looking at the top of the head, and you see these horseshoes. That's the tomoshvari. And there's one right there, and there is one right there. Under a very simple, so the number of ommatidia, remember, this is more similar to what we saw with the entognaths, not the ectognaths, not the insects. So this is a paired sense organ that's likely a hygroreceptor, so telling the organism something about how moist the condition is. And not all lineages may have it. So this is one case where this is the only lineage, the myriapods, where we see the tomoshvari organ, but not necessarily all of them have maintained it through time. And this is a common thread. Again, through the evolutionary trends with all of the major groups that we spent time with, we see evolution as a process not defined by the accumulation of complexity, but often by the loss of something that's unnecessary. So when we think about the chylopods, you are probably standing or sitting, if you're sitting at home, or even if you're on campus, as you watch this, you're probably not very far from a chylopod, even now in the wintertime, because we have lots and lots of them that we've moved all over the world. And one of the most resilient ones is this Scutigera species, this European house centipede, that lives in probably most of all of the basements around the city of Guelph. Lives in moist environments. They have 3 pairs of mouth parts, and they have the forcipules, which are right here. So this is an adapted maxillipad, so part of the mouth or one of the appendages associated with the segments at the anterior end. And these are poisonous in that they can be injected into, and sometimes quite poisonous, so that even large prey items like us, for a very large one, which maybe we'll get to some examples as we go along here, you can require hospitalization. It can really hurt. They have these anal legs back here that are not associated with running or moving, but they're predators. They're very fast. If you picture these guys kind of scuttling across the floor as you flip the light on in the middle of the night, and lots of similar segments, and flat. If you were to slice one of these things, they look like a pita bread as opposed to a millipede, which if you slice it in cross section looks like a loaf of bread that is risen. Diversity that's all over the world. There's a couple of great videos that the BBC has that probably, I'm going to talk about them. I'm going to show one of them, and I'm not going to show the other. So I'm going to not show the life in the undergrowth, because that will get this video flagged by the BBC. But there's a QR code that I encourage you to go and watch this incredible video in the Life in the Undergrowth series, where Attenborough was visiting with centipedes that lived in caves, where the centipedes are as big as your forearm and predate bats, hang from the ceiling and grab onto bats. watch the video. There's other lots and lots of this diversity living in the dark, in the moist, is found for both centipedes and millipedes in the dark. And so here's a small, so this isn't the same taxon, but here's another clip of a slightly nervous TV presenter reconciling his nerves with seeing centipede diversity in caves. Was taken to hospital when one of them bit him. So I hope that came through, but please scan either of these just in case. Their integument, their chitin is pretty permeable. And so, again, this is another adaptation or a requirement that they live in moist environments. And again, the spiracles can't, that's what this kind of cross-section is here, into an atrium and into the trachea, but they can't close this kind of ravenous mouth that is the parent in the spiracle here in figure B. They're Malpighian tubules. They have them to excrete the metabolic byproducts like, and they believe that this is actually another case of the independent evolution of Malpighian tubules from the hexapods. They produce uric acid as waste, and they can actually live a long time. Remember this number next time you flip a log and you see them in the forests on campus. 4 to 6 years some of these predators can live. So there's a long blind ending diverticulum of the gut that is about kind of concentrating metabolic wastes, and they are hosts to a lot of things. So not just predators, but hosts. And since this is a link to thinking about when we were talking about entomophagy last week, this is, I suppose, a reminder that that's like eating anything. It's not without consequence. And so you want to make sure that you cook your centipedes if you eat them. This is a story that from a medical journal that the New York Times picked up a bunch of years ago about a mother and son that had rat lungworm in their brains after eating wild-caught centipedes raw that the son had purchased at a market. And you've run into rat lungworm before. It's a nematode, Angiostrylus cantonensis, that can lead to these two kinds of meningitis in humans. And usually it's transmitted by worms. Sorry, by snails. These are round worms. Just as an aside, this is the species of centipede that they ate. And so we've got a very simple compound eye here, like 6 ommatidia there. Now the diplopods, that's the time that we have for the centipedes. I love the millipedes. This is a millipede from probably, this is at the top, this is cloud forest at the top of Volcan Cacao in northwestern Costa Rica. And I love the coordination of these diplo segment, the legs bearing apparently paired sets of legs. Millipedes are nocturnal detritivores with a hard or mineralized cuticle that actually involves some calcium salts that they accumulate from the environment. And the rings that you see here or here are diplo segments. And so that's a segment, like 2 separate segments that during development fuse. So they are 2 segments that lose some of that segregation. So each diplo segment, the 2 segments that's fused together, bears 2 pairs of legs and 2 pairs of spiracles. So it gives the appearance of being of paired legs, and it might have something to do with coordination. They are not predators, and they have an enormous number of adaptations associated with their repugnatory glands. They are a chemist of productivity, all sorts of anti-prey chemicals, including methylclone, like a sedative that can, and hydrogen cyanide. So quite toxic things. But here's, for those of you who have a chemical bent, here's kind of a menu of the different chemicals that might be released from the openings along each of these diplo segments. So 8 different molecule types, including as a hit parade at the top: phenols, hydrogen cyanide, quinzalones, alkaloids, benzoquines. And the quinzalones, this includes a synthetic type of this compound is methylquin, or a quaalude, which was a potent sedative that we built for humans and was used as a recreational drug in the 60s, 70s, and 80s. And if you watched Scorsese's Wolf of Wall Street, you might remember this scene where DiCaprio's character has drugged himself or been drugged and is trying to get into his rather cheesy automobile. This was a quaalude. And so some of the hypotheses that these repugnatory glands, the range of compounds that are protected or released in them, they might actually affect predators in somewhat of this way, to slow them down, to make them fall asleep, to allow the animal, the millipede, to escape. There's a couple of different views here. There's a type 1 and a type 2 and a type 3 gland that is associated with each one of these, so like an epidermal invagination in each one of these diplosegments. So we've got at the top here with this type 2, since it's kind of the most iconic one, we've got a valve muscle that opens up, and this opening from the integument, that's this kind of lined area here, follows, the valve opens, it's like a stopper, follows a duct and into an area where there is hydrogen cyanide. And so in type 1, this is a type 2, this is type 1. Type 3 polydesmid millipedes are actually highly resistant to their own secretions because they convert them to innocuous chemicals. Here's a type 3. So there's a reservoir where they secrete a stable precursor. And then instead of just a stopper to the outside world, there's a valve, a vestibule, where there is an enzyme that is secreted, and only when the two are mixed does the active form of the chemical become produced. And these are really potent chemicals. So a 1 gram millipede has been estimated to be able to produce 600 milligrams of hydrogen cyanide, which is more than enough to kill a person. So don't eat these. They are often very honest advertisers of their toxicity. So they're often aposematic. They're often very colorful, and there's a whole lineage, the polydesmid millipedes. So they're slightly flattened, and they have kind of these plates that come out on the lateral sides. And this is a suite of species in a group in Appalachia. All of them produce cyanide. All the species are blind. All the species are colorful, and their distributions within the Appalachian area is sympatric. So I'm going to have a question about these. So we're going to bring up the timer in a second. But this question, where all of them are producing cyanide, all of them are colorful, and all of them are living sympatrically. So in that photo array, there's two species. There's species that live sympatrically, species from each genus, and don't worry about the phylogeny, but all of them produce toxic hydrogen cyanide. So which of the following statements is false? And there's only one. And I'm going to put on the timer. You can pause and figure that out. I'll have a drink. Okay, so I'm back. I'm sure you figured it out. And it's B, right? So Müllerian mimicry is when there's kind of this species, a between species congregation around this honest signal, the bright colors. Batesian mimicry is when there is a model that is honestly signaling its toxicity, and then a mimic taxa that has the same colors but has no actual toxicity. And so here's where these pairs of species tend to live within some area of the Great Smoky Mountains. Cylindrical, so if you cut them in cross-section, they look like that loaf of bread. Only 2 pairs of mouth parts, one of them called the nathalorium. You can see them in 2 different drawings here and here. They are the most diverse of the Myriapod taxa, more than 10,000 described species, and many, many more to be discovered and described. They're cryptic, and they live in dark and hard to get to places, so there's a whole bunch of taxa that we just have not bumped into yet. So that lower lip, the nathalorium, it's involved in it has a whole raft of kind of chemosensory and tactile receptors. Thinking about that diversity, the undescribed diversity, I'll give you a couple of recent examples where, for example, this is a new species of polydesmid millipede that was common in one city park in Tasmania, south of Australia, but is absent from any of the eucalyptus, any of the forest groves just outside of the city. And they described this species knowing at that point that it had a known range of 12 square kilometers. Many things to be described and discovered. This is another amazing group. The genus is commonly called a dragon millipede that has these aposematic colors if they're found above ground, so they're brightly colored, indicating their toxicity in the raft of the chemistry that they can discourage their predators with. but there's many of them that are less colorful that are distributed in caves in and around Asia. These are 2 species of dragon millipede that were described from Laos. And then, of course, I used to, when I took this course and when I started teaching it, talk about the fact that in truth there's no true millipede. There's no millipede that has 1,000 legs or more than 1,000 legs until a few years ago, 60 meters underground in Australia, the first true millipede with 1,306 legs was described. And this is a look at that long, beautiful, exquisite creature. Now, in Canada, we have a lot of them, but like a lot of fauna in this recently glaciated area, about half of these have been more recently introduced by our own activity. So about half of the 48 species that we know of in Canada are introduced. So in addition to the chemistry that they use to discourage and the honest signals that they have in terms of their bright colors, there's also some incredible millipedes that were relatively recently rediscovered in California. And that is another polydesmon. See, these are the plates I was telling you about. So we've got kind of paired legs on each diplosegment, these false segments, but the plates, they're paired legs. And this is the unanticipated discovery, like the unanticipated discovery when we start shining UV lights at things, that this species is bioluminescent. Now, here's a look at its closest relatives here in this phylogeny, and you can see the size of the circle is kind of a measure of the intensity of the luminosity of the bioluminescence, and the fact that they all have it. So it seems to be a monophyletically derived trait. This is a single clade that possesses this trait. And if you look at the signal, the size of the intensity of the luminosity, it seems to grow brighter through evolutionary time. And researchers think that this luminescence might have initially involved a chemical pathway that was designed to deal with metabolic stress by living in a hot, dry environment, and then repurposed as a warning signal, because one of the other things, these species are kind of isolated on higher mountaintops. And some of that may have been associated with a greater predation risk. So kind of, there's a lot of cool things that remain to be discovered with millipedes. And there are millipedes all over the place. This is a forest floor that you can click on and explore later on if you'd like, in French Guiana, so the northern Amazon, where there is a millipede right in here. And if you're interested, there's another where there are millipedes all over the leaf litter here in the northern part of Algonquin Park. And I'll leave it to those are kind of Easter eggs for you to go and look for if you're interested. Now, for the rest of the time together, we're going to talk about crustaceans. Crustaceans, this is the Tribbiani clade, right? Famously not a monophyletic group. So we can either talk about crustaceans as being the insects of the sea, but there's a lot of marine biologists that would flip that on its head and say, no, 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 no. Insects are the crustaceans of the land. And in fact, this group, Pancrustacea or Tetraconata, has been hypothesized is maybe for all of the panarthropod groups. So just hexapods alone it's certainly the driving force of diversity. But if you were to expand it to say insects being crustaceans of the land, so essentially Tetraconata or Pancrustacea, that by far and away becomes the most biomass and the most diversity in any of the phyla of animals. There are some apomorphies that would support this, synapomorphies that would include it. So Omatidia with 4 crystalline cone cells, and this group would essentially be this comprised of the hexapods and the crustaceans. And there's some mantis shrimp and some fly eyes showing their apparent the synapomorphy of having this very complex eyeball. So many thousands, tens of thousands of species of crustaceans in terrestrial, freshwater, and marine environments around the world. That's a joke there, aka also known as food. These are critical taxa to the ecology and the functioning of many systems around the world, particularly marine system, because much of the world eats crustaceans, plankton. A lot of the plankton is comprised of crustaceans. So the cool things that comprise this group, we'll think about 2 pairs of antennae, 2 pairs of maxillae, that they have a compound eye, Saccate nephridia. This is involved in their getting rid of metabolic byproducts, toxic metabolic byproducts. Simplified tagmata, a head and a trunk. The trunk appendages are biramus, so those are the legs that have kind of 2 ends, and one of them might be about walking and one of them might be about feeding currents. All the trunk segments have appendages. This is different than the insects, where only the thoracic segments have appendages. Suspension feeders use mixopods in a ventral feeding groove, and they have a specific larval type called Anopolis, a Anopolis larva. So lots of synapomorphies, but a very unresolved phylogenetic perspective of the group. Here's from chapter 21, here's all the characteristics of the crustaceans that you can go through later on. And just a dominant presence in environments around the world. So there have been estimates that there are about 1.3 times 10 to the 21st copepods. So this is like thousands of quintillions of copepods in the oceans, assuming that there's only 1 per liter, which is a vast underestimate. They have some of the largest invertebrates and some of the smallest, and they live parasitically, terrestrially, marine, and freshwater. So food. This is a view from earlier this week when the Sebastian was still sampling in the South Pacific Ocean, just north of Antarctica. And when they turned on their lights, it was very hard for them to see a lot of what they were sampling because the water was filled with krill. Because krill, they grow and they support an enormous ecosystem in the South Pacific Ocean, the South oceans at the bottom of the world. I'm going to take a stab at this, and if it doesn't work, if I have to, what we're going to look at is a small clip from the frozen planet where the importance of krill is described.
Krill. They have been grazing on the algal layer throughout the winter. As the ice melts, more of the algae are released into the water. Both algae and krill flourish in the summer sun.
Look at those bellies, full of algae.
The krill collect the algae in the sieve-like basket formed by the interlocking hairs between their front legs.
So let me go back here for a second, because I want to show you just... So you're watching in that video, or I hope if you've been able to click on it and watch it later on, you see that the legs at the bottom of the abdomen, at the posterior end, they're moving, they're swimming. But the legs at the front, near the trunk, near the head, are essentially a fine basket in which they're moving, but they're not really moving there to move the animal. They're filtering. They're interlocking little hairs on their appendages that they're using to filter and then move all of that food up and into their mouth. So like I said, some of the largest, some of the smallest, so approximately a millimeter long. So from a Reynolds perspective of whether the world is viscous, this is a viscous thing, but as soon as you're bigger than this, you're into the inertial world, and then you think, well, here's a decapod that can be 4 meters across and 44 pounds. So 4 meters across is like that's its leg span, but that's a car, right? That's a big invertebrate. So here's Crustacea, the non-monophyletic Crustacea, and a nod to just some of these taxonomic names, some of which you've run into before, some of which you might not have, none of which is really, memorizing any of them is really the focus of what I want to spend time with. But we've got incredibly iconic groups here like barnacles and Daphnia. We've got groups that are less well known like the brancharians that can be ectoparasites, tadpole shrimp, which sometimes you may have actually had experience growing, decapods, so things that we eat like lobster and shrimp, but also including things like woodlice or isopods, copepods, and then not very diverse groups like the Remipedias, where there's only 10 species and they live in freshwater caves, and morphologically they look like they're ancestral crustaceans. They look like what we would imagine that an ancestral crustacean would look like. And then there's the behavioral diversity, the ranges of diversity. Again, this is a course, but what I've chosen to focus on that here is that there is a deep sea species of crab in the Atlantic called Paramola caveri. And it is known, it was known from collections when we used to dredge them, but as soon as we've been able to send ROVs down to take high resolution video, what we found is that these things run around, not with their claws, but with one pair of their legs, and they hold up live sessile animals or shells dorsally above their head, and they're just holding them there. And we don't know what they're doing. So these are two pictures where they're carrying sponges. Hypotheses are it doesn't seem to be food. As we've watched them, this is still a developing technology, so we don't have a great idea. But they don't seem to be eating them. It may be about camouflage, but go out if you're a marine and freshwater person and you're looking for a really interesting taxon. Get on some ships and come back and tell us the story about the carrying behavior of the deep sea crabs. Now, eating, as we talked about briefly with the krill, is often about adaptations associated with the legs. So suspension feeders like those krill would be using mixopods. So mixopods are a ventral food groove, which is over here. So you can see the arrows moving from right to left as my mouse kind of tracks it across. So there's an exopod here that might be involved in movement or respiration. Then an endopod that's about kind of moving those around. And here's a look at it in cross section. So we've got exo and endopod in the food groove in the center. So a mixopod would be a leg that is a combination of walking and or leaf-like portions for swimming. So we've got some legs that are being used clearly to walk and some legs that are being used to swim, and all of those kind of associated with generating food, moving up to the anterior and into the mouth. Freshwater examples. There's one of the most dominant animals in aquatic systems are zooplankton, and they're involved in some of the, like all of these crustaceans, involved in essentially the largest migratory event in the world, the diel vertical migration from up at night to down during the day. And these are a critical part of food webs in lakes and ponds, and they can achieve enormous densities. Teaching a field course at Churchill on the shores of James and Hudson Bay in northern Manitoba, this is the density of Daphnia in some of those pools that are within sight of the ocean. So, like a lot of arthropods, their success of these crustaceans is related to specialization associated with segments, but also with articulation and their appendages. So the apomorphies of them of the arthropods in general, say if it's about segmentation and articulation, in the crustaceans it's really about the articles. So we've got two very simplified tagmata, the head and the trunk. There's the head, and we've got all sorts of appendages associated with each one of these segments. So we've got, lots and lots of antennae, first and second, on this little shrimp-like fellow here. And then there's feeding mandibles and pairs of maxillae inside of the mouth, and pleuropods associated with feeding and or movement, and parapods associated with walking. So one of the writ large evolutionary trends within the crustaceans is this fascinating transition from being a pelagic swimming organism to a benthic and or terrestrial organism. So the story of, and within the decapods, this most diverse group that has lobster and shrimp and krill and is one that goes from pelagic, from swimming to crawling. And so we see like a shrimp that is much more swimming oriented than a lobster, that is much more swimming oriented than a crab. and switching the images here to focus on a couple of different regions. The biggest change isn't actually, even though there's very simplified tagmata, the head and the trunk, is that the biggest change here is in the movement away from this kind of powerful escape mechanism of the abdomen as the swimming form in the shrimp, less so in the lobster, and then tucked right out under the body. So in a crab, you've moved this crab body plan has essentially become, through time, in many different lineages independently, generally wide and flattened with an extended carapace. And it's moved from being something that's neutrally buoyant to negatively buoyant, so it's not floating, it's walking. And its direction of mobility has changed at right angles, and it often has become, as you've seen with crabs, walking laterally. So carcinization, there's an actual word in evolutionary biology, is to describe the evolution of a crab-like body plan that has evolved at least 5 times and been lost at least 7 times, and is associated with multiple invasions of the terrestrial environment. So the pleon, flattened and bent, so blue here is essentially the crab-like shape. And you can see across this phylogeny that there's an appearance of the crab-like shape with a non-crab-like sister taxa at least 5 times. Why? Well, some of the advantages might include reducing the surface area vis-a-vis the abdomen that's exposed to attack that nice juicy muscle of the lobster that we love eating, of the lobster and the shrimp. Well, what if we tuck it under and away? That the locomotion means that sideways stance means it can essentially escape in any direction with equal speed, and roboticists and biomimetically inspired engineers are interested in crab walking often for that very reason. And then the focus on the chelae, the advantages associated with having these large claws might be about opening up new niche space in terms of who's eating whom, having becoming more capable predators upon other invertebrates. They're gonochoric. There's males and females. They fertilize internally. Many of them use a spermatophore, and the sperm duct here is used by males for the indirect transfer of sperm to the female seminal receptacle. The sperm tends to be not mobile, and there's a whole bunch of terrestrial species that use visual and auditory cues to bring this relationship much closer to ensure that it is going to happen successfully. In the myriad of environments, from parasitic to terrestrial, aquatic, and marine, and then within the marine environment, we find crustaceans, we find crabs nearly everywhere. That includes at deep sea hydrothermal vents. And you might have heard of this one, the Kiwi puravida. So this is a yeti crab, which is found at deep sea hydrothermal vents off the Costa Rican shoreline in the 1990s or the early 2000s. I love this species because it's gorgeous, and because its species name, the epithet, is a contraction of this, in Spanish, a Costa Rican phrase, pura vida, which means pure life, literally, but is often just an expression of joy and of good health. They have these, it's called the yeti crab because of these highly modified hairs that appear all over the body, and these waving arms. So these hairs are actually a place to grow the bacteria that is the main source of food for the crab. So they're not eating anything except the bacteria that they grow on the hairs that grow on the outside of their body. And then they have these highly modified third maxilliped, so like this part of the mouth that can then harvest the bacteria. And that's some of what you're watching right there. So there's the third maxilliped, and you can see it kind of grooming the hairs off of its arm. And they're famous for being observed by the ROVs for waving. And the hypothesis is that the waving might remove boundary layers, static water that's kind of built up around those hairs or around their appendages, that might limit the capacity of the bacteria to fix the carbon and then therefore the growth. And so then they're one of my favorite taxa, because not just because they're found in one of my favorite nations, one of my favorite places in the world, but because that rhythmic arm waving has led to their somewhat limited internet fame, because you can set those to music. These are the Yeti crabs discovered and described off the Costa Rican shoreline. That's good stuff. Let's talk about isopods for the last few minutes that we have together. This is a already dead alligator that is being put down by scientists and weighted down at the bottom to observe decomposition and the decomposers from the marine environment. This football-shaped thing here is an isopod. What's an isopod? It's a little pill bug, a sow bug, a potato bug. There's all sorts of common names, none of which mean a whole bunch, but I offer all them all to you so that you can picture. It's that little trilobite-looking thing that's underneath the wet logs or the wet stones in your basement when it's like this long. But in the marine environment, they can be this long. And some of you know about this species. Here's one that's available for sale for consumption in a market. But one of them was recently discovered in the deep sea environments off of Vietnam and was named after Darth Vader because of its face. They figured it looked very much somewhat like the face of this iconic fallen Jedi. They are a really characteristic organism and food for much of the predatory insects in the tropical rainforests and cloud forests that I work. And this is amazing. This is the isopod. This is an amphipod, which we're going to put aside for a second. But the isopods have a whole bunch of adaptations associated with their movement back into the terrestrial environment. So in spite of the fact that their cuticle is relatively permeable, they can survive. So you can see here, this epicuticle, right here, very, very thin. So they lose water very easily. But they have a number of adaptations that prevent that water loss. Some of them are the fact that they have pseudotrachea and gills. So let's take a look at the fact, the pseudotrachea, so the gills are exposed, there we go, there's a gill right there, associated on the interior of the leg. So there's the leg with the maxillipad and the inner carapace wall. And so that's a gill, but the pseudotrachea are better at conserving. So this, in a marine or a freshwater environment, this is a great way of breathing. But in a terrestrial one, it's not. And so they have pseudotrachea, where here is the attachment to the leg. Here is another part of the leg, the ecto and exopod. Here's the spiracle that leads into these branching of the pseudotrachea. So there's a spiracle that goes into a pseudotrachea inside of the interior of this crustacean. So an adaptation, and we're using words, a pseudotrachea, almost a trachea, the same adaptation that we saw in the myriapods, that we see in the insects, that we see in a lot of the terrestrial arachnids. And the pseudotrachea for this crustacean are these protected invaginations that are better at conserving water than our gills. And they can tolerate, the taxa that live terrestrially that have pseudotrachea can tolerate much drier air than can those species with gills. And this seems to be, again, a trachea as a convergent thing. They don't possess all of these different lineages, don't possess the trachea because of shared ancestry. Now, one of the other cool adaptations they have, this historically marine organism for living terrestrially, is something called a marsupium. And that is a protected pouch right up here, nestled just in the interior end of the body. And it's a brood pouch where the females put their eggs in their first instar larva and where they rear them in this protected area that begins, and they're kept moist, essentially. They keep their babies in the water, and then they finish in the final developmental stages in the air. So it's an example of extended parental care as well as an adaptation to a terrestrial environment. So pseudotrachea, and the marsupium as being some incredible things to think about the next time you roll over a log or disturb an isopod in your basement or in your woodlot. Alrighty, here we have been talking about centipedes, millipedes, and crustaceans. And the next time that we're together, we're going to be talking about some of the results of your facial mite survey in lab and some of the previous years when we were able to sequence some of those facial mites. So until then, I hope the weather is something that you can enjoy. I hope that you're doing things that are good for your mental health. So take care of you, take care of those around you, and we will talk soon.